Okay, we're back. In this part of the so we're going to starting fresh right here. We're going to start with the pentose phosphate pathway. So this is the alternative pathway to metabolize glucose. It is accomplished by the pentose phosphate pathway to PPP. PPP does not oxidize glucose via glucose 6-phosphate to produce ATP. Instead, this pathway produces specialized products such as NADPH. Looks very similar to NADH, which is required for numerous reductive biosynthetic pathways and provide pentoses for nucleic acid biosynthesis, including the production of other monosaccharides such as all of these sugars, which are required for the synthesis of other important biomolecules in humans. So the significance of the pentose phosphate pathway. So besides ATP, these cells also need reducing power of NADPH for biosynthesis, anabolism. Just remember, if you think reducing power, reduction, oil, rig, reduction is gain. So it's going to be giving stuff, so we're going to be building. Whereas NADH is used for oxidative metabolism or catabolism. If you think way back, breaking stuff down. So the cells keep the remember these brackets mean concentration. So the concentration of NAD plus and NADH ratio near 1,000. So that means that we typically have 1,000 to 1 ratio of NAD plus. This favors metabolic oxidation. The cells keep NAD plus and NADPH ratio near 0.01, so very, very different ratio comparing the two. And this favors reductive biosynthesis or anabolism. So oxidation of glucose by the PPP generates NADPH. The PPP is also known as the hexose monophosphate shunt. So if you see HMP shunt, it is the exact same thing. These are just AKAs. You will have test questions have all of these different names. Also the phosphogluconate pathway, all of these are the same, literally mean the exact same thing, just AKAs for one another. So don't let that trip you up. So the pentose phosphate pathway also generates ribose 5-phosphate, which is required for nucleic acid biosynthesis. So without the PPP, we would not have RNA and DNA being made, and RNA is needed for all of our protein expression, and DNA is who we are. It's needed in all of our cells, otherwise there are no cells. The cell cannot function. So we're going to look at the different parts of this pathway. So the first part consists of oxidative reactions. So glucose 6-phosphate is oxidized to 6-phosphoglucono. I'm fairly certain that that is the delta sign. Let me confirm. Yes, that is the lowercase delta. So this is 6-phosphogluconate delta lactone by glucose 6-phosphate dehydrogenase, which forms the first molecule of NADPH. The 6-phosphoglucono delta lactone is converted to 6-phosphate gluconate by 6p gluconolactone hydrolase. Oxidative decarboxylation of the 6p gluconoactone by NADP forms ribulose 5p, CO2, and the second NADPH. Focus heavily on when these certain things are created. Those are typically going to be the most important steps, and the ones that most likely you'll get asked questions about. So the 6-phosphogluconolactone dehydrogenase catalyzes the synthesis of a second molecule of NADPH in this reaction. So both glucose 6-phosphate dehydrogenase and 6-phosphogluconolactone dehydrogenase enzymes which produce NADPH, use NADP plus as the electron acceptor coenzyme, and magnesium ion as the cofactor. Magnesium ion. You'll learn later on just how important magnesium really is. It really is a cofactor in a lot of different reactions, and it's often one that many people are deficient in because it is only found it is found mostly in like those dark green leafy vegetables and other vegetable type sources that have higher concentrations of these good metals. Okay, the second part of the PPP is that the ribulose 5P produces ribose 5P and xylulose 5P by phosphopentose isomerase. Thus, the ribulose 5-phosphate, a ketopentose, is converted to an aldopentose ribose 5-phosphate. If you think way back earlier when we talked about sugar and we talked about ribulose versus ribose, we had that chart to help us keep some of those straight. So this is all coming back full circle. And the ribose 5-phosphate is used for nucleic acid biosynthesis. Be careful not to get those two mixed up on quizzes and exams. Ribose is for the RNA and DNA. Ribulose gets converted to ribose. The third step, the non-oxidative phase of the PPP, involves trans-ketolase and trans-aldolase. So we have in the keto and the aldo, the trans meaning transport. So we're going to be transporting these groups. The trans-ketolase and the trans-aldolase reactions require thiamine pyrophosphate. Remember that we've seen that before. It's our TPP as a coenzyme. In tissues that require NADPH for reductive biosynthesis of important molecules, both the pentoses, ribulose 5-phosphate and ribose 5-phosphate, are converted to glucose 6-phosphate by a series of reactions. Ribulose 5-phosphate is epimerized to form xylulose 5-phosphate by ribulose 5-phosphate epimerase. Ribose 5-phosphate can react with the xylulose 5-phosphate to form cetoheptulose 7-phosphate, followed by its conversion to fructose 6-phosphate and then to glucose 6-phosphate. The later reaction that converts ribose 5-phosphate to cetoheptulose and then to fructose 6-phosphate and glucose 6-phosphate are catalyzed by trans-ketolase and trans-aldolase, enzymes which are requiring the thiamine pyrophosphate TPP as the coenzyme. This kind of repeats itself twice in this whole paragraph, but these reactions with the oxidative and non-oxidative phases of pentose phosphate pathway are shown below. So see the glucose 6-phosphate, 6-phosphogluconate, we get our NADPH, and then we get the ribulose 5-phosphate. These trans-ketolase and trans-aldolase can convert us back to glucose 6-phosphate. And then we'll keep going, so we keep continuously to produce that NADPH. Or it can continue on to go into ribose 5-phosphate and give us the nucleotides, coenzymes, DNA, and RNA. Okay, the three 5-carbon sugars, ribulose 5P, ribose 5P, and xylulose 5P form fructose 6P and glyceraldehyde 3P by trans-ketolase and trans-aldolase and enter glycolysis. So this is also a way that we can take, like if we need energy, we can take ribulose and form glucose in order to produce additional ATP as well. So they got another step there as well. So not only do we get to form NADPH, but we can throw this back, create glucose, and throw the glucose into glycolysis to make more ATP as well if we need more ATP. Okay, this right here, this is going to go over the specifics. The trans-ketolase is going to go over the specific reactions. Trans-ketolase enzyme catalyzes the transfer of a 2-carbon moiety comprising carbons 1 and 2 of a keto sugar to the aldehyde carbon of an aldose sugar. As a result, the donor ketose is converted to an aldose with 2 carbon less, and the acceptor aldose is converted to a ketose with 2 more carbon atoms. Reaction requires thiamine pyrophosphate as a coenzyme and magnesium ion as a cofactor. For example, xylulose 5-phosphate reacts with ribose 5-phosphate to form 2-7-phosphate, and glyceraldehyde 3-phosphate. Literally repeating again exactly what was said above. This is really repetitive. Transaldolase catalyzes the transfer of 3-carbon moiety dihydroxyacetone from the ketose cetoheptulose to the aldose glyceraldehyde 3-phosphate to form ketose fructose 6-phosphate and tetrose rethros 4-phosphate. And here is our nice chart of our pentose phosphate pathway, the different steps, going step by step. This whole process to create, getting that ribulose. Remember that ribulose. Hold on one sec. I think there might be a typo here. Yeah, I think this is a typo. So we see here, so we get the 6-phosphogluconate converts to ribulose. This should down here should be ribose 5-phosphate, not ribulose. Ribose, little typo there. Thought that looked weird. Okay, we have the oxidative reactions of the pentose phosphate pathway. So this phase of the pentose phosphate pathway generates NADPH and ribose 5-phosphate. The ribulose 5-phosphate produced in this phase is used for transketolase and transaldolase reactions to finally form glucose 6-phosphate from pentoses as shown below. Again, a whole lot of pictures, all this stuff. This is literally just a exact repeat of everything that we just read. Okay, don't focus so much time on these pictures here just because there's so much here. Understand the main concepts. Understand transketolases, kind of what those are, what those do. Transaldolases, what they are, what they do. Understand the ribose and the ribulose, and I would make sure you are familiar with that it goes from G6P to the delta and then to glucose 6 G6G, then to ribulose, then to ribose. Okay, break it down a little bit easier so you don't have to freak out by looking at all of this stuff. Okay. Again, this is giving you each, again, most individual step by step of things that are going on, that are happening. Just take some time to you can shorten these out a little bit. If again, understand the different pieces of it. Like if you understand that xylulose and ribose are just epimers of each other, meaning they have the same chemical structure, just a slightly different shape, or same chemical composition, but just a slightly different organization of those chemicals, then you understand the isomerase, epimerase, that's going to be what's going to happen. The fructose and glucose, same thing, these are epimers, so they're isomers of each other, so you're going to have an isomerase. Okay. Now the significance of the pentose phosphate pathway, this is really the meat and potatoes, what you want to know for this unit. So NADPH production for biosynthesis of macromolecules, the ribose 5 production for nucleic acid biosynthesis, and when ribose 5P is not needed, you result in 2 NADPH per glucose 6-phosphate. When ribose is needed, the ribulose forms ribose, and then all this gets converted to the ribose 5P by reversible reaction. So we get, when it's needed, we get the ribose. So the utilization of glucose 6-phosphate produced in this pathway depends on present time cellular needs. Availability of sufficient NADP plus that may result during conversion of NADPH to NADP plus. In some reductive biosynthetic pathways can direct glucose 6-phosphate to be fed into PPP. In contrast, when the demand for NADPH is low due to more oxidative pathways requiring energy, glucose 6-phosphate enters glycolytic pathway to yield energy. So this is showing when we have a need for a whole bunch of energy. So let's say we're exercising, running around. our body's going to convert and take all this stuff and can push it into glucose 6-phosphate and push it into glycolysis. If we're in more of a relaxed state, kind of like that rest, rebuild, like create catabolic versus anabolic. If we're in more of an anabolic state, like building that muscle, recreating all the stuff that we need, then we're going to be converting more into the ribose and doing our thing there. Okay, just make sure you differentiate between those two. So clinical correlation. This is a really good one. Wernicke-Korsakoff syndrome. You will get asked a question about this. Wernicke-Korsakoff syndrome is caused during severe deficiency of the vitamin thiamine. The active form of thiamine is thiamine pyrophosphate, TPP. So if we don't have thiamine, we don't have thiamine pyrophosphate, we don't have TPP, and the pentose phosphate pathway cannot be performed because we need it. We need TPP. Where's the... Yeah. We need it in order to do the transketolase and transaldolase enzymes. Have that reaction occurring. So right here, getting between ribose and everything else, we need that TPP for these two reactions. In this genetic disorder, mutation in the transketolase gene causes a defective enzyme synthesis having very low binding capacity for TPP. This low binding affinity of TPP to transketolase decreases the operation of the pentose phosphate pathway. So, with Wernicke-Korsakoff, this is one condition that pops up so it can't, in the book, in his case, he talks about it being a genetic disorder mutation. More often than not, you see it's the lack of thiamine, so it's caused by the vitamin deficiency. The vitamin deficiency typically is a result of diet choices, especially with alcoholics. So alcohol actually will prevent the body from absorbing thiamine, or vitamin B1, same thing. And you'll start to see a lot of these issues when you don't have TPP. And actually you also have a lot of memory and brain type deficits, as this can have like a severe effect on the brain. So when you think Wernicke-Korsakoff syndrome, it is in some cases a genetic disorder mutation, but more often than not it is due to alcoholism. And that is all of chapter 12. Did it in two parts. Had a little bit of emergency with baby, with my baby in there in the middle. But hopefully it'll be nice to have it broken down into smaller videos, that way you can tackle them individually. Okay. Have a great day. Remember, keep studying and work hard, and I wish you the best.